Chapter 1 of Microbiology. Microbiology is the study of organisms that are too small to be seen with the naked eye. This means, we're going to have to use a microscope. These organisms include bacteria, viruses, fungi, protozoa, helminths, and algae. Bacteria we normally think of as this. Viruses, most people would think of as microbiology. Fungi fall into this category because some fungi are single-celled organisms, so they're very small. And we throw them into this group. Protozoa are all single-celled organisms. Helminths themselves are not necessarily single-celled. They're not single-celled organisms, but we usually throw them into this group because they're small and they cause diseases. So they just get thrown into this group because of that. And then algae, and these are the single-celled plant-like organisms because they technically are not plants, but they are very similar to plants. They kind of fall into the Protista. They're in the Protista kingdom along with protozoas. Jobs that fields of microbiology include public health and epidemiology. So these are tracking diseases. You hear about it a lot in the news. You have environmental microbiologists. The big thing with NASA on these is they are searching in extreme environments to see what life is there and how to test for it. So that way, when we send probes to another world, we know where to have an idea of where to look, where we might be able to find some kind of microorganisms and what kind of studies, tests to do to maybe discover microorganisms on another planet or celestial body. Right up here in this category, corner, we have biotechnology. Biotechnology is how we use microorganisms. So we can use them to make acids or some kind of a chemical. We can use them to make bread, wine, cheese, beer. All of that stuff is microorganisms. There should be biotechnology. Creating ethanol is biotechnology. In the lower right-hand corner here, you have immunology. Immunology is the study of the effects or the reactions that our body has when we have an infection. When a microbe invades us. So that's what immunology is. In the upper left-hand corner here, you have genetic engineering. Genetic engineering is where you take a gene or some kind of a trait from one organism and you put it into another one. A very beneficial example of this would be the gene for producing insulin. We put into E. coli so that we can produce more insulin cheaper, so that people who have type 1 diabetes, who are insulin dependent, have more access to it at a cheaper rate. And that is a positive. Nobody complains about that form of genetic modification. We also do food and things with it so that we can feed everybody. Agricultural microbiology, on the other hand, is maintaining the soil for the plants that we grow, so that way they thrive better, they're healthier. A fictional example of this would be the movie The Martian with Matt Damon, where he's growing potatoes on Mars and he uses his own species to do this, and then they can't do it afterwards because we need microbes for plants to grow. Most plants have some kind of symbiotic relationship, some way, shape, or form with microorganisms. And then they also studied the diseases that affect plants that we like to eat. Food microbiologists, these are the ones that try to maintain clean environments for our food, so that way we don't get infections. They do a pretty good job of it considering the amount of food we make, and we do hear about contaminations every now and then, but as food microbiologists, it's their jobs to try to minimize that as much as possible. There's a list of other things underneath it, but those are the main ones, main categories. The origins of microorganisms. They've been around for most of the Earth's history. The oldest fossils ranging from 3.5 to 3.8 billion years old. Cell types, we have two main types. We have prokaryotes, which are our simpler ones. They're smaller. Then we have our eukaryotes, which are bigger. They appeared much later in the geologic history of the Earth. The key thing here, pro means pre, karyote means nucleus. These are before nucleus. And then U means true. So these are true nucleus, these cells are. There's a time, the oldest fossils are in this area here, out there quite a bit. Eukaryotes did not even appear for more than 1 billion years afterwards. So by far, prokaryotes have been around for a long time. So again, prokaryotes, all microscopic because they're all single cell and they lack a nuclei. That is very important. They lack a nuclei, okay, and membrane organelles, but we see it because of the nuclei. So eukaryotes, on the other hand, these are unicellular, and we have multicellular ones. But the big thing here is that they have a nucleus, and we can almost always see that nucleus, and they have membrane-bound organelles. Viruses, a big one, most people, almost everybody's heard of, are a little different. They have a little bit, because of this feature right here, they have the A in front of that cellular. Anytime you see the letter A in front of a word, that means without. So viruses are without cells. So technically, viruses are not alive. All right. We say they're active or inactive. But a lot of people will misuse the terms live or non-living or dead instead of active or inactive. They are different than cells in the fact that they have a nucleic acid and they have a protein coat around them. Over here, you see this, but they don't have all the other features of cells. So they're considered non-living. Microorganisms are important for the flow of energy and the flow of nutrients or matter through the environment. Energy comes in the form of light from the sun. There are far more cyanobacteria and photosynthetic microorganisms than there are plants, and they produce at least 50%, if not more, of the Earth's oxygen. So they convert light energy into chemical energy that gets passed through the ecosystem, and it leaves the ecosystem as heat. All right. They also turn inorganic matter, carbon dioxide, into organic material. Now, if it wasn't for the microbes, the bacteria, and fungi, we wouldn't have any decomposition because those are our two main decomposers. And they're the ones that take that organic material from dead things and turn it back into stuff that other organisms can use. So that's where it cycles through. So they bring energy in for us to use, and they keep the cycle of nutrients and matter in the ecosystem. Again, how do we use organisms? Biotechnology is using this. So we use it to produce food. Heck, they're even growing steaks in labs right now made out of bacteria. They're pretty expensive, so nobody's doing it yet, but that's a possibility in the future. Most of our antibiotics come from microbes. Right? We grow vaccines from these things. Again, producing ethanol, bread, cheese, those things, that's technology. Manipulating these is the genetic engineering portion, where we take something from one thing and put it into something into another organism, and it works because we're all related to each other. Again, we talked about the insulin. We do this with food, with fruit. Anybody like papayas? But if we didn't have GMOs, we couldn't have papayas anymore. I don't care what anybody says. All the papayas out of Hawaii that are grown there are genetically modified organisms. And a fairly relatively recent, relatively speaking, bioremediation. Bioremediation is where you use microorganisms to clean up waste products. The most common form of this is wastewater treatment, where we use microorganisms to remove the excrement and other waste products that are in the water and clean the water up. So dirty water comes in, clean water comes out, and that's how we can keep our environment cleaner and keep fresh water for us to use. Most microorganisms are free living, don't cause us any problems, so we don't pay a lot of attention to them. Minor field, some fields do that because they want to know more about it because it could be beneficial somehow. Those that do live close with us, sometimes they're parasites, and that means that they live on or in us and that they're causing us some kind of a harm. Now, it doesn't mean they're going to kill us, nothing that bad, but they're causing some kind of a damage to a host. We are going to refer to humans with this most of the time. So that's what a parasite is. It lives on or in, and it causes some damage to the host. Pathogens, on the other hand, are ones that cause diseases, which is different than the parasite, slightly different than the parasite. We have about 22,000 different microbes that cause diseases. We have 10 billion new infections a year throughout the world, even though there's only a little over 7 billion people in the world. So that means people get more than one infection. Pretty simple. 13 million people die every year from infections. Most of these infections are simple ones that can be treated with antibiotics or just clean water. And that's the biggest thing. A lot of them are in poor sanitation areas, and that's why the diseases get transmitted a lot through there. So worldwide mortality, influenza, you get a few 100,000 people to 500,000 people that die per year from influenza alone. Typhoid, measles, something we take for granted in this country. Worldwide, not just in the United States, but worldwide, you get over 100, close to 150,000 people die from that each year. So, and most of these things we can treat or prevent. We have emerging diseases. These are newly identified diseases. Between 1980 and now, we have roughly 90. There's a hot button one, yeah. COVID-19 is a emerging disease. It's a new one. That's why they call it a novel coronavirus. Zoonosis. This means these are diseases that come from animals that normally live in animals and doesn't cause problems with the animals, but it jumps the species and comes over to humans. And because it hasn't evolved with us, it causes us problems. And that's kind of what's going on with COVID-19. Re-emergent diseases, these are older diseases that with sanitation and with medical practices has kind of gone to the wayside, and they became really scarce, but they're starting to pop back up again. A good example of this one would be measles, actually. Measles should be eliminated in this country, but because people do not get their measles vaccine in this country, they travel outside the country, or we have tourists who come in or immigrants who come in who have not been vaccinated from it and they could be carrying it. We see it popping up in this country again because of people who choose not to get vaccinated, and it's easily preventable. Most of the time it doesn't cause any problems, but it can kill. There are people who die from it every year, even in this country. And then we have our neglected tropical diseases. A lot of these is something like malaria is neglected. We could prevent a lot of it. We can cure it, and we can prevent most of them, but we neglect them. We can prevent a lot of malaria just by having mosquito nets. But poor countries, they can't afford them. We take it for granted because an average mosquito net's probably $3 to $5, but you're talking like a month's salary for a lot of unindustrialized countries. So again, here's some other ones. Schistosomia. Most of these are parasites that get in from, again, easily treatable, but it's from unclean environments, and people are exposed to it, so it gets transmitted. All right, we've had thousands of microbiologists over the last 300 years because microbiology really is only about 300 years old. It couldn't start until we had microscopes, so that's why. So we couldn't see it. We didn't know they existed. And then we have a few things here, and we'll go through those in the slides. So before microbes, in about 150 years after microscopes were discovered, created, we had what was spontaneous generation. And this, excuse me, well, even after microbes first were discovered, spontaneous generation, this is where people thought that organisms just, boom, appeared out of the air, out of thin air. Like if you put manure, boom, you're going to get flies. The flies are going to grow from manure. They didn't lay eggs and have maggots or anything to turn into. And that was later disproven over several years, several decades, as a matter of fact. Pasteur was the last one to finally disprove it, and he came up with the theory of biogenesis. And people don't question this. And what this is stating is that all new life is only coming from existing life. So we don't get, you can't take some seeds and some burlap and throw it into a dark corner and boom, that's going to create mice. That just doesn't happen. The seeds will attract the mice, and they'll use the burlap to build nests and stuff, and that's how they get there. But they used to think that that's how mice were actually created. All right, now our first microbiologist is Anton von Leeuwenhoek. He did not create the first microscopes, but he was around the time when they were first created. He did make his own microscopes. They were single lenses ones. And he was the first to view living organisms. And he was fascinated by them, and he drew them all the time. He made lots and lots of microscopes. After this, we started using the scientific method even for the spontaneous generate to disprove spontaneous generation. We had to kind of formalize it. People started doing it. Okay, well, this is a problem. How am I going to solve this problem, or how am I going to explain this? And that's what scientific method is. So it starts. You see something. You form your question, your hypothesis, which is your educated guess. All right, it's the best answer you can come up with what you know. So then you design an experiment to test that. So then you run your experiment, you analyze it, and you get your results, and then it may or may not agree with what you thought, and that's okay if it does not. Most science, in this case, gets it wrong because it doesn't support what they thought it would. They thought it was one thing, and it turned out to be something else, and that's good. We learn something. That's what the process is all about. But again, the hypothesis is the best guess, or the best explanation they can come up with. All right. As you go through this, once you do it, you publish your ideas. Then other people will look at it and they're like, "Oh no, you made a mistake here," or they repeat it. It's like, "Oh no, you did a good job. It came out right." But it gets repeated by others. So that's where it's what we call peer review. If your hypothesis stands up to this, and other people's hypothesis support that also, then you can go up to a higher level of knowledge on this, and a higher level of confidence, excuse me, and we call this a theory. So the theory has been tested many, many, many times over years by lots and lots of different people, and all their different experiments has come back and support this idea. That's this phenomenon. That's what a theory is. If a theory is even more compelling, then we call it a law or a principle. In biology, we don't have a lot of these, but we can have some, all right? But we just don't we put more of them into physics or in chemistry. So here's an example of scientific method when studying diseases. Edward Jenner noticed that because smallpox used to kill people, it had a 30% mortality, far higher than COVID. But he noticed that milkmaids would get a disease called cowpox from cows, and it only affected their hands, and they would stay pretty. All right, and they would never get smallpox, which was deadly. Cowpox wasn't. So he came up with the idea of taking some scrapings from their sores, and with willing participants, we couldn't do it this way now, but he did have willing participants. He took these scrapings and he made little cuts on their arms, you can see, with the scrapings, and put it in there. Well, these children ended up getting cowpox on their hands, which is fine. It would, again, it's not dangerous. And then he would later try to infect them with smallpox. and they wouldn't get it. So he's the one, we give him credit for modern vaccinations because of that. People were doing stuff similar to that hundreds of years before him, but he documented all, so he gets credit for it. This is our first case of true vaccinations, and because with his method and everything, we no longer have smallpox in the wild. You don't have to worry about catching smallpox anywhere unless you work in a couple of labs throughout the world, just a couple of labs in the world, because smallpox was eradicated in 1979 in the world. Thanks to him, because vaccination does work. All right, we discovered some features about microbes. John Tyndall, and Cohen discovered that some microorganisms were immune to heat. I'm going to put immune to heat. You could boil the water or boil the broth or whatever to kill everything and let it sit there, and then they would still, grow. So they looked at these microbes under the microscopes and noticed that they had little structures on the inside of little spore-like things, so they called them endospores. And endospores are a way for microbes to resist harsh environments, okay? And from that, we determined that we needed to have, we get this sterility. And sterility means we got to destroy all of our the viruses that are around there, all of these spores and anything else. It's this high level of cleanliness. It's hard to maintain, actually. But we, especially in certain environments, we try to do our best with it. And that's all you can do. That's what sterility is. They did some stuff. Wendell Holmes and Ignace Semmelweis noticed that mothers getting sick and whatnot for those who gave birth in hospitals, or from those who, when doctors would go from an autopsy table to a maternity ward without cleaning themselves, noticed that people would get, the women would get sick more often that way than otherwise. They started making that correlation, and then Joseph Lister was the first one to, building on their information, it was in a short amount of time, relatively speaking, they came up with, there's some microbes that are being transmitted this way. We take it for granted now, but they didn't know that back then. And he came up with what we call aseptic techniques. And aseptic techniques is washing your hands, putting on gloves when you're touching things, trying to prevent contamination. They used to not do that all the time because they didn't think anything about it. So using chemicals or sterilization, or heat for sterilization, to try to prevent these things happening. And we still have some contamination nowadays, but we're much, much better than we used to be. Honest, much, much closer to a sterile environment than we used to be. All right. So with the scientific method, we have some big theories here. The germ theory of disease is a major one. And this one here probably could be called a law, because absolutely nobody questions this, and it's been proven over and over and over. But in the past, we used to think that people used to think that if you got a disease, if you were sinful, or if you were a bad person, or because you were poor, you got diseases. We later found out it's because of sanitation and other stuff like that. But at the time, a lot of it was because you were sinful. That's why you can even read in the Bible that people who were sinners got diseases, were sick until they were healed, and then the disease was gone from them, and then they were no longer sinners at that time. They were born again, or however you want to say it. The two main contributors to this here is Pasteur and Koch. All right, Pasteur started, he's a little bit older than Koch, but they were working on it around the same, they both were working on it while they were both alive, so they both get credit for it. All right, so Pasteur came up with it when he was working with the fermentation in the French wine industry. They, the wines were getting spoiled in their vats, and he came up and he figured it out that it was because of a bacterial infection in there, and you had to heat it up and clean them out after each use. And we call that now pasteurization, named after him. Then he's famous for the disproven spontaneous generation, proving that it was transmitted from one organism to another. So because of identifying that it was microbes that were causing these diseases is where he gets his credit for the germ theory disease down here at the bottom, is because of that. It starts again with the fermentation. Pasteur is the one, Koch is the one who finalized and finished what Pasteur started. He did these with he came with postulates, and that was to I'm going to use, say, a lab rat that's infected with a disease. You isolate the microorganism out of that lab rat, put it into a healthy one, and that healthy one should get the same disease, and then you can re-isolate that same organism out of that second lab rat. And that's how they proved that it was an individual type of bacteria or individual type of microbe that was causing a specific disease, and not sin or bad character or something like that. To do this, though, he had to come up with this bottom portion here, the developing the pure culture methods. And he was the first one to do that. So he's the one who definitively proved the germ theory that Pasteur had started. Pasteur didn't have pure culture methods until after Koch discovered how to do some of these, and then other people started doing it. So they both get credit for this. He's the one who identified anthrax, tuberculosis, and cholera, and he made vaccines for most of these, not all of them. He and Pasteur were big into vaccines also. Another thing, we have to classify organisms. So taxonomy is the science of organizing these material and putting them into groups, okay, and then naming them so that no matter where you are, if you get the scientific name, doesn't matter what language you're speaking, it's going to be the same. Carl von Linné, or he's also known as Carl von Linnaeus, he's got a few different names he's known for. All right, so this is the orderly arrangement of these organisms. They gave them nomenclature, so they started assigning them names using, you had to use Latin on this, and they had some parameters that you had to use, so that was consistent for everybody. And they did this to record traits, to determine where they are related to other organisms. In the early 1990s, we came up with domain. We have a lot of these other, most of you probably have this now. But we have domain. There's 3 domains, archaea and bacteria. These are both prokaryotic domains, and eukarya is only eukaryotes. There's 5 kingdoms. Then those get broken down to different phylums, different classes, different orders, different families, different genus, different species. All right, so here's an example of this. So these are all eukaryotes, both sides of it, doesn't really matter which one we go to. These are animals, even sea squirt and sea star are animals. Then if you go to the phylum Chordates, you got a little more narrow. You had to get rid of the sea star. Go to mammalias, you had to get rid of that sea squirt. Go to primates, had to get rid of the canine. Go to hominids, you had to get rid of the lemur. Go to the genus Homo, you had to get rid of the gorilla. And then there's only one genus of Homo surviving to this date, and that is Homo sapien, us. Assigning the names, we come up with what we call binomial nomenclature. And binomial means it has to have 2 names. All right. The first name, the genus, is always capitalized, and the second name, species, is always lowercase. When you, if you write, you can write these in italics, like right here, it's Staphylococcus aureus. If you do not write in italics, you must underline them. Each word gets its own underline also. And the first time you write them in a paper, you always have to write the whole name out. After that, you can abbreviate the genus. You can come up with all kinds of different names for these things. There is a process to get it approved, but you can still come up with names, quite colorful names for some of these things. Phylogeny is how these are related to each other. We used to do this by the way they looked, thinking that that was how they related, but now we use genetic markers to show the relationship, and that's how we ended up with domains that changed the other classification process. So this is all based on evolution, again, showing how they're all things change over time. New species originate from pre-existing species, okay? And then the more closely related they are, the more similar features they're going to have. And as they get less and less related to each other, they're going to look more and more different. And that's all this is talking about right here. That's what evolution is. Here's the kind of a tree of life for it. We have the monarans down here on the bottom, and this is the domain of bacteria, the true bacteria, what normally people think of when they think of bacteria, and archaea. And archaea are ones that live in extreme environments where it's like high heat, high salt, either low pH or high pH. Yeah, those are just ones off the top of my head I can think of. Could be high pressure for that matter. All of these above this dotted line are the eukaryotes. So you have your kingdoms. Again, the 5 kingdoms are Monera, Protista, fungi, plants, and animals. All the Monarans are single-celled. All protists are single-celled. The fungi are single-celled and multicellular. All plants are multicellular, and all animals are multicellular. So this is one way to show them. I prefer this next one a little bit better. And it shows how animals kind of broke off over here to the side, and they put them by relatedness. So animals are more close, or excuse me, eukaryotes are more closely related to the archaea than they are to bacteria. But notice that there's a trait right here from the bacteria coming over and joining to the eukaryotes. This first one represents mitochondria, and the second one represents chloroplast. Okay, so these are, and back here at the beginning, these ancestral lines, there was a lot of interchanging and mingling, so it gets really convoluted, and it's over time how they disperse is where we get all of our different types of organisms now. And that is it for this chapter. We'll get the next chapter coming up soon. Thank you.